Good morning, everyone. It is indeed a great pleasure to welcome all of you on our last day of this 5-day international workshop. I hope everybody had a great time being here. So, moving on, we have a few announcements for our participants. First of all, for this hands-on session, I hope everybody is ready with their accounts made on UseGalaxy. Secondly, we will have our validatory session at 3:30 p.m. IST. I request everybody to please fill the feedback form correctly for your e-certificates to generate. Thirdly, I request the participants, if they can, please mail us a short video of explaining how was their experience of the conference. We would try to play it at the valedictory session. Thank you, participants. Now, may I request Dr. Yasha Hasija, coordinator of the workshop, to please introduce our both experts for today, Dr. Anshu Bhardwaj and Mr. Sangram Sahu. Ma'am, please.
A very good morning, everyone, to the last day of the International E-Workshop on Bioinformatics. And trust me, this day is going to be no less, neither in terms of information nor in terms of energy. And I am pleased to introduce you all to the session expert, Dr. Anshu Bhardwaj, Senior Scientist, CSIR-IMTECH, Chandigarh, India, Assistant Professor, Academy of Scientific and Innovative Research, India, and Team Lead, Drug Discovery, Virtual Tool Room, Drug Discovery Hackathon 2020, Government of India. Dr. Bhardwaj, a biologist and data scientist, obtained her PhD in 2008 in understanding genotype to phenotype correlations in human mitochondrial diseases from CCMB, Hyderabad. She then joined CSIR as one of the founding PIs of the open source drug discovery project in 2008. She conceived, designed, and implemented crowdsourcing as a tool to tackle challenging scientific problems in the Connect to Decode project. She has over 40 publications to her credit and has trained over 1,000 young students. She served as an associate scientific advisor to Science Translational Medicine and is on the editorial board of Frontiers in Systems Biology and Journal of Genetics. She has several honors and awards to her credit. She was one of the 4 innovators in India, United Nations Development Programme, UNDP, selected for the prestigious International Visitor Leadership Program by the US State Department, awarded Newton Bhabha Fund from the British Council and the Royal Society of Chemistry, UK. More recently, she was also selected as the first female long-term CRI fellow, Paris, France. Her passion is biomedical big data analytics with focus on understanding infection and rare disease biology towards better therapeutics. She has conducted several workshops and has been invited to deliver over 150 talks at national and international platforms. Today, she would be presenting this session with Mr. Sangram Keshari Sahu, Bioinformatics researcher, UseGalaxy India Outreach Team, Bioclues. Mr. Sahu completed his MSc in Bioinformatics from Pondicherry University in 2017 and then went ahead for his research on gene regulatory networks and role of long intergenic non-coding RNAs at early stage development in plants using high throughput sequencing data at IISER Mohali. He is interested in open and shared platforms for doing research, and along the journey, he has contributed to open source projects such as the Data Carpentries, UseGalaxy, NF4, SnakeMake, Bioconda, etc. He has developed several tools for genomics communities such as SigBio Shiny for performing gene enrichment analysis and Arabidopsis EcoGEX for comparing gene expression across different accessions or strains. As a part of Open Life Science program called OLS2 Cohort, he is writing an online handbook on practical guide to reproducibility in bioinformatics. Now I invite Dr. Anshu and Mr. Sangram to take over their session on data-intensive science and need of web-based workflow systems for ease of sharing protocols and results, examples from Galaxy. Over to both of you.
Great. Thanks, Dr. Yasha, for having us here today. And I'll just start by presenting my screen so we can go forward. So can you confirm if you can see my screen?
Yes, ma'am.
Oh, great. So thank you so much, Dr. Yasha, for having us for this workshop. So as you can see, the title is pretty elaborate. So I thought I'll simplify it for ease of reading as well for understanding what we are doing today. So the first thing that we need to understand is that the need for bioinformatics or computational biology is in the first place lies in the fact that we are in a data intensive world. So the talk is focused on data intensive science and what is the need for web-based workflow systems which allow us for ease of sharing protocols and results. And I hope by the end of the workshop you will understand what we mean by that and why it is so critical in a data science like in a data science scenario to make sure that the protocols and data are shared in a way which maintains transparency. We hope that myself and Sangram will be managed to convince you that why do you need to use these systems, and we'll take you through one hands-on example today and one example which we will just do a demonstration so that you know how these platforms operate. Now, before I proceed any further, I'd like you to take a look at what is on the slide and think what it is. What do you, and if you have any ideas in terms of what do you think it is, you may type it in the chat box. So this is the lab notebook of Graham Bell, and it is from 1876. Now, in order to do science in a reproducible fashion, or in order to report what you're doing, imagine like so many years ago, if you were thinking logically, this is what you would logically do. You would have a page number on your lab notebook. It should be dated properly. It should be written in waterproof ink and not pencil so that you cannot erase and rewrite. There is an entire description of the procedure. There the observations are listed, and whenever there is a mistake, instead of striking the mistake completely, you are just crossing it so that you can still read what the mistake was and can follow it up. The reason I'm showing this lab notebook is because this is a classical case of ensuring that when you document something, when we document an experiment, these are the minimum set of information that one should document, and nobody practically taught Bell to do that. He did it on his own because it sounded logical, right? Now, how do we make sure that when we do data intensive science today, we follow all of this? And if you realize most of this has to do, people who are actually coders here or use some kind of platform will realize that this has to do something with version control. This has to do something with the fact that you can actually trace back and go to the earlier observations that you made. How did you change it? When did you change it? Why? You know? So we actually now will discuss the replicability versus the reproducibility of the work. So there are four terms that we practically use in a very interoperable fashion, and it's not practically the same thing, but any experiment that you do should follow all four. It should be repeatable, which means same experiment in the same lab. It should be replicated by somebody else, same experiment in a different lab. The data that you observe should be reproducible, so it's the same experiment but in a different setup. So it's not that you do a experiment in a specific way to get that result. If your observations are right, even in a different setup, it should show the same result. Reuse. Different experiments, some of the same, which means that it should be modular. If I have taken the whole experiment and it has like 10 steps, one should be able to take step 1 to 2 and should get the same result. So the point is, any experiment has to have all of these four components, and it's very critical that we follow these guidelines when we report any scientific discovery or observation. In order to do that, the scientific community actually came up with something that we understand today as fair data. I am pretty sure the previous speakers for past 4 or 5 days would have referred to fair data, but I would like to re-emphasize on it. So FAIR stands for findability, accessibility, interoperability, and reusability. And the bottom line is that if a data is readable to human and understandable to human, it should be similarly understandable by our computer programs. So the machine actionability or the capacity of a computational system to find, access, interoperate, and reuse data with absolutely no or minimal human intervention is the key to do data intensive science today. If your data which you generate in the lab or you're using from a public domain does not follow FAIR, it's going to be extremely difficult to make the entire protocol reproducible. And reproducibility is the essence of science. So it's practically a non-scientific way of doing data analysis, right? For the simple reason, there is a constant increase in the volume, in the complexity of the data that we see today. So we will very quickly see what do we mean by FAIR. So F here is findable. If you cannot even find the data that you are referring to, you cannot do anything with it. And how do you find data today? You find data by assigning metadata to it. Say for example, my name is my metadata. My actual name Anshu is the data. My age is the metadata. My actual age is the data. So every data set has to be assigned a metadata. And as you might have observed by now, the metadata identifier should be standard. Everybody should be saying name and mean the same thing by it. I cannot say it's something else and somebody else is talking name something else. Like say for example, locus, gene name, ORF. You know what I'm talking about. You have the same name, the same meaning, but different identifiers for the same data that you will see. And metadata or data are assigned globally unique and persistent identifiers. When we say that, it means that if I'm using an identifier today, it should not vanish tomorrow. I should be able to follow it up. And of course, the other things are more of a follow-up on that it should be very rich in metadata, it should have unique identifiers, it should be indexed and searchable somewhere. So that is the whole purpose of keeping data findable. Accessible. Once the user finds the required data, it should be accessible. Like if I want to retrieve something, what are the communication protocols to retrieve it? I'll give you a simple example. If I don't have the mobile number which is in 10 digits and you start typing 9, the service provider keeps on telling you this is not the right mobile number, you cannot connect, right? Which means that the 10-digit mobile number is a communications protocol. Now, how do we define those communication protocols for access of data that is out there? So that is free and universally implementable protocols in place. And when we take the hands-on in Galaxy today, you would know that how these platforms allow you to generate such protocols. And every protocol, this allows for authentication and authorization processes wherever necessary. And of course, they should be interoperable, which means that data need to interoperate with application or workflow for analysis. A very simple example again, if I share something which is in a format that can only be accessed by a Microsoft Excel and no other application can open it, it becomes a problem, right? But the minute I have the backend data in a format that it actually is amenable for several applications to work on it, the more and more people can actually use it. So it is important that the data has to have interoperability, and one of the most important thing in interoperability is the vocabulary. And when I say vocabulary here, I'm pretty sure some of the previous speakers would have talked about the gene ontology, the dictionary by which we call everything, and that is very, very important. And coming on to the last part, reusable. And I think we already saw this. Reusability is simply the fact that if I have used something and I have a 10-step process and I want to use 6 to 7 to 8 steps for something else, I should be able to reuse that. So it should be modular. And if it is not following FAIR, it won't fit in the scheme of new events that you are proposing, right? How do you know if you have followed the FAIR data principles in your project or not? There is this website which is fairsharing.org, and it defines the minimum information for biomedical data analysis, okay, biomedical investigations. And if you see here, it has this information for natural sciences, life sciences, engineering, computer science, informatics, biomedical science. So if you go here, you would find the resources on data and metadata standards, which allow you to make your experimental data follow the FAIR principles, and that is very, very critical. Now, since we have understood what FAIR means and why everything has to be reproducible, let us see how do we do that. So today's focus is to basically work with Galaxy. As I understand, one of the speakers yesterday did inform you about the Galaxy workflow system. So I need not go through details of what it is, just to tell you it's a workflow system where you have these three components. The left side is the list of tools. In the center, you have the canvas where you see how your tools are getting connected. And this is history. So as and when you are running an application, what you have done gets captured in the history on the right side here. And once you are done with the analysis, one can extract these workflow from the history to say, okay, I have done 100 steps, but I would like to use only 70 of them for my workflow system. Those 20 were exploratory in nature. And so basically, we'll show you how Galaxy works today by taking one concrete example. We'll take up a biological question, see what kind of data is required to answer it, and let's see if we have answered it. And then we'll see one more example, all right? Going forward, I was not sure how many of you really have the idea of or the concept of what a workflow means. So for those in the audience who would like to know what it is, so a workflow simply means this. Now this is a conveyor belt, and on a conveyor belt, what you are making is or assembling is a pizza. So if you see here, there is a dough which is placed on the conveyor belt, and then there are three pans on the top which are programmed to add a specific amount of sauce, cheese, and mushrooms to this pizza dough, right? Now imagine if you manually do it every time, the amount of sauce or the cheese or the mushroom will not be the same. But now if I program how much of sauce and how much of cheese and how much of mushroom has to be added, every pizza that goes out of this conveyor belt will exactly be the same, right? Reproducibility, exactly same things. But if you do these steps manually, it will not be the same. We know that. The problem that translating the same concept in a scientific scenario is that in most cases, our workflows are more complex than assembling a pizza. So then the problem happens is that manually you can still get your pizza problem solved, but if you now need to translate that same concept in a scientific situation like this one which is shown here, the problem is if I change a decision making somewhere here, or if I try and change a route here, I may have to redo a lot of other connections. And this can be a mess. So in order to simplify this, people actually started designing workflow systems to basically perform routine, extensive, complicated tasks without any manual interference, just for reproducibility. You can share the methods and protocols as it is, and there is a lot of transparency. And we will actually show you when we say transparency and sharing of protocols, what it means. You all might have read a scientific paper at some point, right? There is a specific section there which says materials and methods. No amount of detailing there will actually tell you how those materials and methods have been really used in real life. So if you take a publication today and you read materials and methods and try reproducing it, do that as an exercise. It's a very good exercise for the young students out there, and you will now then realize how hard it is. Sometimes you won't find the details of exactly what was done here, because the minute you start dealing with the data, you realize, okay, I don't have this information. The paper doesn't say anything about it. So then you start assuming. The minute the assumption seeks in, you know that this method is not reproducible because I'm getting slightly different results. Now that slight different result can have huge implications in terms of the interpretation of the whole experiment. So that is very, very important. Even more challenging is the fact that genomics data will, it's projected to be 1 zeta bases per year in next 4 years. So if you see here 1 zettabyte, what it means on the slide, so you know how much data are we talking about. And then it's only the genomics data, the raw data. Then comes the annotation. Then comes the interaction data. Then comes your networks. Then comes your metabolic modeling. So you can imagine how mind-boggling will it be to actually analyze this data in a way that one group has done the analysis, but the other group is struggling to reproduce the same, right? And there are several methods that people are using in this context, like machine learning, artificial intelligence, network science, predictive modeling. All of this cannot be reproduced if you don't put this in context of a workflow environment. And when I say that, I mean that you need to transfer this into something which we understand technically as a pipe architecture. What does it mean? So, like say for example, you started with an input 1. Say for example, I have given the raw sequence of a newly identified organism. Okay, and I don't know how many genes it makes, how many proteins it makes, what it does. So I just sequence the organism, and that raw data becomes my input 1, and my job 1 here is to identify how many genes are there in that raw data. So just trying to put punctuation marks, saying, okay, this is gene 1, gene 2, gene 3, gene 4. So define the structure of the genome. Now, once you have defined it, in output 1, you would have the structure of the genome, right? Now, that output for the first job becomes input for the second job. What is the job 2? Now I know where are the genes. My second obvious question would be, what is that they are doing? And once I know what they are doing, then I can start understanding how do they interact with each other, how do they bring a phenotype, what kind of genes actually code for protein, which does not code for protein. Then you actually start building this workflow in your head. And the advantage is now what you are thinking in terms of a workflow, that thought process can actually be translated into these steps of events. And this is a pipe architecture where you actually use a starting point and keep generating outputs at several occasions, which becomes input for your next job, right? And as I can show you, these are custom design workflows. So nobody has to decide for you. You decide what workflow you want. You decide in what order you want to do that analysis. And that is the reason of this being that every analysis that you are doing is modular. So if you would like to do step 3 before you do step 1, that is a decision that you have to take depending on what kind of biological question you are addressing. And not just biological, it can be chemistry, it can be material science, it can be anything practically. So the applications of these workflow systems are not restricted only to biological sciences. Anywhere you have data that is available, it will work. So the point is, it's modular, it's reproducible, it's shareable, and it's scalable. As you can see here, 1 to 10 genomes, it doesn't matter. And the last point, which I've really not written here, but people who really cannot code need not struggle to see, they're not dependent on somebody who can really automate these tasks for them now. You can go to a workflow system, it's a drag and drop activity. So you really need not understand to do command line coding anymore. There are so many platforms that allows you and makes it easy for us to work on it. So practically what I'm trying to convey is that there are several research labs across the world who are working on generating a lot of data, generating a lot of methods. And the point is that every other researcher in the world should be able to reproduce what each of these labs does in exactly the same way as it is reported. And the Galaxy is one of the platforms that allows you to actually do that. Give you a very brief introduction. This is the project page where you will find a lot of information on where do you arrive at Galaxy servers. There are several Galaxy servers now in the US, in Europe, and in Australia, and Africa. So there are many Galaxy servers. You can have an account on any one of them. And there are several public servers of Galaxy where people have customized their own application for Galaxies. Now, if you see here, one of the top applications here is AB Open Lab. It is an application developed by my group. So the point is, I was not finding these applications on Galaxy, so I simply had a local version of Galaxy at my end and integrated the programs that I thought would be useful by the community and shared it back on the Galaxy platform. So you can use the central resources or you can develop your own resources and pool them in a central fashion. Now, this is all about the fundamental principles of what fair data is. Why do we need a workflow? Which workflow is good? Why are we using Galaxy? But today, when Sangram gives you a hands-on, I would like you to really understand the context behind it. So the problem that we have identified today to give a workshop demonstration is antimicrobial resistance. And there are obvious reasons for it. So I'll actually very briefly tell you what AMR means, so that when Sangram takes you through the workflow system, you have the context in your mind, okay, I'm trying to solve this problem, and this is how you can use Galaxy to solve one aspect of that problem, okay? So in 2017, the World Health Organization identified a list of these priority pathogens. The reason for identifying them was that these pathogens are basically causing havoc in clinical settings. None of these pathogens are easy to treat. So they are all showing a very, very stringent, resistant response to different antibiotics. And I'll show you what I mean by that in next few slides. So if you say antimicrobial resistance, so the definition goes like this. It occurs when microorganisms such as bacteria, viruses, fungi, parasites change the ways that render the medications used to cure the infections they cause ineffective. Now, to simplify this, if today I could treat a X bacterial infection with an antibiotic A, but this X bacteria mutated and converted into something which is X dash, this antibiotic does not work on this bacteria. As simple as that. So an antibiotic which was functional earlier is now no more effective. And when a microorganism become resistant to many antimicrobials like that, we start calling these organisms as superbugs, because they are now have identified multiple ways of avoiding multiple antibiotics. Okay. To give you a more visual example, in a population, sometimes you actually already have, we actually don't realize, like bacteria really don't have faces. So you cannot say this bacteria is different from the other one. But in a population, we know that there will be heterogeneity, right? So sometimes in a population, you already have an antibiotic resistant organism. And the minute you give antibiotic, as you can see in the middle panel, all the susceptible strains die. So what happens is what remains and what now replicates are only the resistant ones. So sometimes by over-prescribing antibiotics, we are driving the evolution of eliminating everything which was susceptible and promoting bacteria which are more resistant. And that is why this problem becomes very intense, because now if you don't use antibiotics properly, this, and of course we are not using it properly, and that is why we are seeing this problem of antimicrobial resistance causing havoc. All of us are impacted by the pandemic, right? If you go out there and look at the number of publications in terms of why AMR is a bigger problem than the ongoing pandemic, you will realize what I'm talking about. SARS-CoV-2 is just one pandemic. The AMR pandemic is going on all across the world, and it's like a slow tsunami, as somebody said it. So it's sweeping a lot of lives away. It's just that we don't realize it because we are not treating it as a pandemic so far, but it is actually one. Okay. A more real-time example is this. So what Harvard University faculty did a couple of years ago, they wanted to demonstrate, okay, we are saying that bacteria evolve. Can we visually see how it is happening? So what they decided, they did this mega plate experiment, microbial evolution and growth arena. And what they did, if you see on the slide here, I have had this huge Petri dish, which is 4 feet by 2 feet. So imagine almost the size of a dining table, that big a Petri dish. And then had media on top of it. And then they added a blue color to the media so that the media looks darker in shade. And if you see here, then they started dividing this Petri dish into multiple panels. Okay. The panel on both ends of the Petri dish is 0, which means that there are no antibiotics there. The minute you start going from the periphery of the Petri dish to the center of the Petri dish, you will see it is 1 times the antibiotic, as in this concentration is enough to kill the bacteria. So this is 1 times the MIC, minimum inhibitory concentration. 10 times more dose, 100 times more dose, 1000 times more dose. Okay, so now you would actually have no antibiotic and then increasing concentrations of antibiotics as you go towards the center of the petri dish. Then what they did, they actually plated bacteria on the outermost panel, and as obvious, the bacteria which was growing here could be seen in white in color because we have a blackened agar, like the blackened media. So what you now have is this. So you will see that E. coli appear as white on the black background. And you see, it was obvious that the bacteria will grow where there is no antibiotics. But slowly, the bacteria actually started to grow where there was 1 times MIC, 10 times MIC, 100 times, and 1,000 times. So within a matter of a few days, not weeks, in a few days, the bacteria could evolve to survive in a concentration which was 1,000 times more than sufficient to kill them. So this basically, it was an eye-opener because now you can see this evolution happening in lab in a matter of days, right? So this is something that this is actually available as a beautiful YouTube video. I would recommend all of you to go and watch it. It actually shows a time lapse, a time series video, and you show that bacteria actually then stop at that interface, but then they start growing and then again stop, but they start growing again. So this is what is happening, and this is obvious because antimicrobial resistance is an evolutionary mechanism. So of course the bacteria have had billions of years to learn how to evolve. Now it is not just in the lab if you see what is going on in the clinic. Now what you see in the slide, top panel in green shows you when these antibiotics were introduced in clinic. Okay, so those numbers are the years. In 1943, we all know penicillin came into existence and we started using it. On the bottom panel is the first time when antibiotic resistance was observed against those antibiotics. Let's again go back to penicillin. Penicillin was introduced in 1943, but resistance to penicillin was already observed in 1940, right? You keep following that timeline and come to, say, for example, bedaquiline. Bedaquiline is one of the most promising antibiotics today to treat MDR-TB patients all across the world. But if you see here, resistance to bedaquiline was already being shown since 2006. So the problem is, it is very, very difficult to come up with a new antibiotic, and it doesn't take very long for that antibiotic to go non-functional. And that is why we thought it was important to give you a hands-on today on trying to understand that if you have a pathogen, how do you identify what is causing resistance, or was it basically imparting resistance to that particular organism? How do we do that today? So even today, culture remains the gold standard for a diagnosis of pathogen. Like say, for example, you take a sample from the patient, grow it on a petri dish, let it grow. If it grows on a specific media under a specific antibiotic or not, is when you say, okay, this is X pathogen, and to these antibiotics it is resistant. Needless to say, it is not going to take hours. It is going to take a couple of days for that growth to come. Okay. So first of all, its culture takes time. And this is the only approach, and it's a gold standard currently in clinic all across the world. Okay. Now, what is the problem? The problem is it leads to many times it leads to over treatment and not the right treatment. There can be several problems. You are not treating the pathogen that is actually there. How many times, imagine for us, how many times we go to a clinician, allow him to take a swab, let him identify what the bacteria is, and then make a prescription? We never do that. We go to a clinician, he looks at what are the overall parameters and makes an objective assessment. I think this should be the infection. Let me prescribe a broad spectrum. Why are we doing that? Second, that impacts the emergence of AMR, and that is one of the major issues. We cannot blame the patient who is suffering, we cannot blame the clinician who is prescribing, because there is no choice. Culture takes time. A patient will not let a doctor wait for 3 days to prescribe something, right? The problem is it's not just time. The sensitivity of these methods is very low, less than 60%, because the biological sample has at times very low number of pathogens. And complex etiology of conditions like sepsis makes it very hard. More than 90% of blood culture remain negative in immunocompromised patients suspected of infection. So you won't even know if an infection exists or not. So you don't even know whether to start antibiotic treatment or not. So the problem becomes more intense. So it's not just time anymore. It actually intensifies further. This is to just give you an example. So when you have a sepsis infection or anybody has a sepsis infection, the first problem is the sites of infection. It's not just blood. It is everywhere. First of all, how many tissues will you isolate? How many microbial experiments will you do to identify which tissue is infected? Second, pathogen. Look at the suspected pathogen list in septic shock. It's more than 50. How many organisms are you going to identify in a standard culture practice way? It's just not feasible. And then this is a classical study. I would really recommend all of you to go and take a look at this paper. It clearly shows that if you look at the y-axis and the x-axis, it clearly shows that at the 0th hour, when a person has gone into basically hypotension, you would see that there is an 80% chances of survival if the antibiotic, the right antibiotic is initiated at that time. Okay? Every hour lost, even if you then do a proper antibiotic initiation, by 36 hours, the patient, you've already lost the patient because survival chances actually grow as grim as 5%. So you know what I'm talking about. In such situations, we really don't have the luxury to wait for a culture test. You need something which is very, very rapid because hours that one has to take a decision on if antibiotics are to be prescribed and if yes, which ones. So you know this situation is really difficult to deal with, and that is why there is a huge interest all across the world to have a method that can identify non-culturable pathogens, because we know that most pathogens we don't know on which media they are going to grow. So we are not even identifying them. So that is a method that we need. As I told you, you need to detect an entire range of pathogens, not just one. So of course, the method has to be comprehensive. It has to be very sensitive. A very, very low load of pathogens should be detectable. It should be specific. It should tell you this strain is present in this sample and this probably is resistant to antibiotic A and B, but susceptible to C and D. So a clinician will immediately know, let me prescribe C and D. I shouldn't do A and B because it's not going to respond to it. It should be rapid. Now we know why. It should work with both dead and live pathogen, because as you might be knowing that in case of TB meningitis, the brain TB, in most cases the pathogen is dead. It's the antigen, the inflammatory response to the antigen that actually causes the meningitis problem in human TB meningitis. So you now know why do you need to identify either it's dead or live, and you of course cannot do a dead culture of a pathogen, you know. And differentiate between pathogen and human cells. That is a problem which is still open for a lot of work, but that is what you need to also do. So do sequencing-based methods offer a solution? And yes, they do. So what they can offer? First of all, you don't need to culture what you are sequencing. So it can identify unculturable pathogens, can differentiate between pathogen strains, can identify new drug resistance mechanisms, like say for example, we detected MEXI in Staph aureus. It is rapid with extreme accuracy. You exactly know what you're dealing with. Okay, so it can actually lead to better informed treatment decisions. And then sequence data can be used to develop specific primer for a more genotyping based assay to provide more complete sequence information and develop more specific field tests, because every time you then need not sequence the whole organism, you know that these are the determinants that if I can sequence this gene or if I sequence these two strips, I exactly know what I'm dealing with. So then your method becomes even faster, right? And has something gone into this? Yes. People have used both the traditional, which is the Sanger method, and the next generation sequencing platforms in clinical microbiology. What is that we observed? That the Sanger method has limited sensitivity for minor variants when present at more less than 15%. Okay, and this has been shown in case of viral infections. Whereas the next generation sequencing methods can detect DR mutations present at approximately 1%. So even when we decide to do sequencing, should we use Sanger or should we use NGS? I think the answer is pretty clear. We need to either use NGS platforms because Sanger sequencing misses a lot of these drug-resistant mutations. And the presence of such variants is critical because it has shown to predict an increased risk for therapy failure. So it is not something which is an assumption. It has been shown that if you don't identify this at a very, very good resolution, it leads to a therapy which will not work. Now, what can we do here? As I was telling you, one can go for sequencing in a way where you only focus on targeted amplicon. Like we know that 16S RNA is a method to differentiate between different species, right? It is still good, but it is difficult to determine a new pathogen. It is actually not possible by a targeted amplicon sequencing to identify new antimicrobial resistant genes. So it is good in a way when you already know this, but it is not good in a way when you are trying to discover something new. It will actually be good to repeat something that you have already observed. So it is not comprehensive. The other way which people have been using in a very, very comprehensive fashion is the whole genome sequencing. When you get something, sequence everything that you have. Of course, it is comprehensive and it can help in detecting new AMR genes. Because had we know what causes, like on what imparts the resistance phenotype to a bacteria, we will not be struggling so much in clinic to treat them, right? So it means that there is a lot of discovery yet to be made in identification of novel antimicrobial resistant genes. Now I'll give you one simple example. This is a classical case of what I'm showing on the top panel is how a typical TB treatment goes. So at the time of diagnosis, as in we assume it's the 0th month, the patient is still sensitive, and you isolate the bacteria and you sequence it. So you know what the sequence looks like for a sensitive bacteria, which means that as of that point in time, it is responding to treatment. But since we know that the TB treatment goes on for 6 to 9 months, are several reasons why the treatment may fail. And despite the fact that you actually prescribe 4-line drugs, at the end of 9 months, the patient actually converts into an MDR patient. Now it is actually showing resistant to these antibiotics. What you try and do, you try and again isolate the bacteria, sequence it. So what you now have is a sequence of a drug-sensitive bacteria, you have sequence of a drug-resistant bacteria. But beyond certain points, sometimes happens that after 20 to 20 months of MDR treatment, the bacteria can still be converted into an extremely drug-resistant pathogen, which is not treatable by even these antibiotics. So you isolate that bacteria again and sequence it one more time. So what you will observe over time, even within the same patient, is that the bacteria is evolving into something on which the antibiotics don't operate. Now, if I have the sequence data from all the 3 cases, one can identify due to which changes in the genome, the bacteria is showing a different phenotype. All right. And over time, by using this data from 1, 10, 20, 100, thousands of patients, one can actually identify a machine learning based method to differentiate, because there can be strain differentiation across individuals. There can be many different reasons why there can be different genome sequences. Not all of them may be contributing to a resistance phenotype. So that is when you apply a machine learning method to identify that can you differentiate if you are given this sample, a pool of bacterial isolates, can you identify their drug resistant status from sequence data? Susceptible, resistant. And then can you predict new DR determinants? And that is where sequencing comes in. And that is where your analysis of this data comes in. Okay. And as I told you, NGS is the platform. What is being very, this is something that everybody is looking forward to. I'm pretty sure most of you either have worked on Minion already because it's a very, very cheap platform. It's very quick. and that is why it is basically meant for point of care testing. People are working on reducing the error rate in MinION, but there are so many advantages of using MinION and clinical centers. So there is a lot of work going on to assess even antimicrobial resistance genes and mutations in clinical care using MinION. So what we thought that instead of taking Sanger sequencing or any other NGS platform, why not today we learn of what do we do when you are trying to deal with a drug resistant bacteria in clinic. How do you take that bacteria, design an experiment, use benign to generate the sequence data and identify AMR determinants. So and because we thought it will be nice for us to give you some data set even to follow up after this workshop is over, we decided to go with an online existing Galaxy training protocol which says antibiotic resistant detection. We will take you through this protocol, but even after the workshop is over, you can continue to do hands-on because it's already available online. So this is your material for the workshop for using Galaxy to determine antibiotic resistance genes. Now, as Sangram will tell you in more details, we are trying to initiate something which is called as a Galaxy India project. So we to just to make sure that people within the Indian community start to, because now we are generating a lot of data, how do we make sure that all these experimental protocols and data sets are reproducible by other members of the community? So before I go forward with the presentation, I would request Sangram to basically take us through uh, the protocol of how do we start by taking nanopore data and progressing further to identify and scan for antimicrobial resistant genes. And we'll use several other programs in the process. So Sangram, over to you. I will stop sharing my slides now.
Okay, thank you, Dr. Anshu. So I'm gonna share my screen. Just give me a heads up, like if you can see my screen.
Yes, sir, we can see your screen.
Okay, awesome. So before we start with the hands-on workshop, basically, so I would like to give you a small introduction, like what basically we're going to do through this. So yeah, so as Anshu said, like so what's the importance of detecting this antibiotic resistance genes, right? So as you know, like even she mentioned, like WHO already declared like AMR is one of the like top 10 global public health threats to humanity, right? So going forward, this is going to be very important to cure. Otherwise, uh, like how things going in current time. So yeah, so alongside, like in 2017, WHO identified 32 antibiotics in which are in clinical development, uh, which are on the priority list of pathogens, right? And out of those, only six were classified as innovative. So there are clearly a lack of like accessibility to quality antimicrobials, and like even that ultimately affecting the healthcare system of throughout the world, right? So when you say like it's the important thing, like how to detect it fast and in a timeless and like very good manner, right? So here comes like a very easy tool of study which is called plasmids. If you're from biology, like you must be knowing that plasmids are the DNA molecules, right? And which are like separated from, which are different from your chromosomal DNA. Like here, let's say bacterial DNA. So these plasmids are known to be carrier of this antimicrobial resistance genes across the tissues, right? So let's give a simple example. Here is a antibiotic sensitive bacteria, and we just introducing to a plasmid which have certain AMR genes, right? Then what happens? Like this plasmid can grow by itself, and it can like create large number of replicates, and ultimately it makes the bacteria a antibiotic resistance, right? So now we got to know like why it's important and how this can be studied, like the tool of study. Now how to do that, like as Ansu said, the thing is, oh sorry, one change. Okay, so yeah, so ultimately that's called minion, but yeah, minions. So yeah, that was a bad joke maybe. So this is the device which Ansu showed you before, just before this on slides. So yeah, if you can simply sequence the bacteria, and on real time you can able to detect the antibiotic resistance genes. So this is a nanopore device, and recently they have been like very successful detecting this antibiotic resistance genes in real time. They have a workflow called WIMP, which is like what's in my pot workflow, which is like on real time it can detect your from a lot of metagenomic samples, it can detect the antibiotic resistance genes, right? Okay, so why are we selecting this nanopore? And the one reason is produce the long rates. And you must be till now, if you're doing with NGS, you must be dealt with this thing called short rate sequencing, right? So those are the short base pair, like 100 base per or 200 base pair sequences, right? But here, why we're choosing long rates? They are very easy and fast to assembly, basically. So, and even like they're very simple to assemble. Even the repetitive genome sequence regions can be aligned very quickly, and they help identify like larger structural variants, right? And here we're detecting for nanopore, but this material like which we're going to do today can be applicable to any of the long read sequencing, right? Okay, so for today's workshop, we're gonna go through some of the sequencing which already been done and deposited in this platform called Zenedo, which gives you a DOI, like it's a citable data platform. So yeah, so let's download them and do the analysis on them on the Galaxy platform. So just to give you a background of the sequences, so if you can see, I just downloaded one FASTA file, which we will do in Galaxy platform altogether. So this is just to demonstrate you. And here you can see a single read can be of very long, right? And sometimes it goes up to 20,000 base pairs. That's why we call them long reads, right? And for today's workflow, like ultimately we need to go through different steps, like which is the purpose of the workflow to end with our antimicrobial resistance genes, right? So what I'm gonna do, we have few input genes, or just you saw the reads of the sequencing, then we're gonna like align them. We're gonna do pairwise alignment, then we're gonna assembly them, and then we're gonna again do the pairwise alignment to increase the accuracy of the alignment. Then we're gonna just do conscious module using recons. Okay, so whatever things you can see in bracket, these are the tools which we're going to use for a particular step, right? And in each step we're gonna do a quality check, so that we know our data is like having integrity at each point, right? So yeah, all these materials can be found in this site, which I can share. Maybe you can get this link in your chat box in the mm, YouTube link which you are using. So if Raj can share this one, that'll be great.
Yes, sir, I
Okay.
shared to the.
Okay. Is it shared, right? Okay. Cool. Thank you. All right. So I hope everyone have a login, like as we mentioned in the description of the this video. If you go to your description, there will be a link called usegalaxy.eu, right? and you can just go there and create one of your account, like just go register here and you can create yourself an account. I already have an account, so I can directly log in. So what's the purpose of this? Like basically, we want you to do along with us in parallel, so that you get a feel like what actually doing the analysis in Galaxy platform looks like, and even like how you can take the advantage of this to share your data in a fair manner, right? Which Ansu just mentioned, like it can be easily shareable, and all the steps and its metadata are detectable, right? So yeah, also like if you guys have any question while going through the hands-on, so I'm going to share a link. You can ask the questions over there so that I can prioritize like which questions to answer basically. That'll be great.
Excuse me, sir.
Yeah.
We can share the YouTube chat box link with you so you can directly look at the questions.
Oh, okay, great, thank you.
We'll post it in the Google Meet, sir.
Okay, great.
Okay.
Okay, so this is the materials basically. Like we covered few of the basic things, and finally we're going to start with the getting our data into the Galaxy. So I hope everyone logged into the Galaxy platform, which is just Galaxy, usegalaxy.eu. Uh, then we're gonna start with a new history. So what is history basically? Like if you're starting an analysis, let's say if you're starting a new analysis, so you can simply create a history where all your analysis steps and data gonna be listed out under here, right? So let's give it a name. So you can give it any name. Now, the first thing is we want to get our data over here, right? So you can just go to here, like, so all these data are saved in the platform Zenodo, like, which is just so new. So this is the platform, and they can be directly downloadable using these links, right? All these links. So these are the same files which are listed over here. Okay. So what I'm gonna do, we're gonna copy all of these files, and we can just go to, so here you can say option, like which is upload or download any of the URLs. So it's gonna give you this kind of box where different methods are there. Either you, if you have files in your local system, you can choose this option. Otherwise, if you have somewhere in your university file transfer server, then you can use this. But for now, we're gonna do fetch from the URLs which we already have, right? So you just need to paste all your links over here, then start. So what it's going to do, it's gonna download all your data to your history panel, right? So it's gonna take some time. Okay, so here one important thing to mention, like for now you are seeing these things as a gray color, right? Which means like they are on queue format, like because these are the public platforms, so you have limited resource, right? So sometimes your jobs, so each step can be considered as a job, and they're gonna be in queue initially. Then once the color changes to this orange, so that means like they are initiating, right? Or they are being processed basically. So jobs are being submitted and they are being processed basically, and it's gonna sometime take some time to download all of them. And once they do, they're gonna change the color to green. That means those steps are completed and they are basically downloaded, right? So it's gonna take some time, like hardly 1 minute, because they are not very big. They're about 24 MB to 50 MB. If
Uh,
you have,
Ram, in the time the data is getting uploaded to Galaxy, you can tell them about how many sequences, how many MDRs have they taken in this experiment. This data is coming from how many organisms?
okay, yeah, that's a good point. So let's consider like from where this data is coming from, right? So they have like sequenced about so where this is being sequenced. So this is sequenced from 12 MDR plasmids. So these are MDR like microbial uh, yeah, microbial drug resistance genes, right? And they're sequenced in the platform which we call like Minion, which you just saw. And after 8 hours of sequencing run, they got about this many reads, like 28,000, 2,87,000 reads, right? From dozen of samples. Like these are the all the samples which you can see, and different with different labelings, right? And they are like done different steps like processing them, like demultiplexing. So demultiplexing is just a step like once you get your data from your sequencer, you just make it in a format which can be seen as a ATCG, right? So yeah, so this is about the data. Let's see if okay. So yeah, in here. So if you just want to see like if the data are what you downloaded as intended, you can just use this view data button, right, with each of them. So now here you can see they are like each reads and followed by the sequence. So if you know the FASTA format well, if you're coming from like bioinformatics background, then you must knowing. So this is the metadata or the name of the read, right? And this is the actual read, which is ATCG. And they when I say they are the long reads, they are actually very long. So like I said, they sometimes can go up to 20,000 base pairs, right? Okay, now we have the data, and all these data are like we can do the analysis to all of the data in a similar fashion, right? So for that purpose, what we're gonna do, we're just gonna combine all this data into a single group, so that it can easy to do all the steps on all of these data together, right? So for that, what you can do, we can do like select all the data. So just go to here, select all of them, then go to the option over here and build dataset list. So there's the option called build dataset list. So you can do just that. And here you can give it any name. Basically, let's say plasmid or all my plasmids. Okay. Yeah. That's it. See, if you see now the plasmids, it's a list of 6 items, right? If you just go here, these are the all the FASTA files in a single group. Now what's the advantage of this? We can, if we're running a single tool, instead of giving 6 files, we can simply provide one this list, right? Then it gonna applicable, the tool gonna run an over all the 6 samples. So these are the small steps, but they are really like turn to be advantage over doing a galaxy, right? So now let's go back to our workshop material. So the first thing is we obtain the data. Okay, so why I'm going through this step by step, so that even if you're not able to do it right now, once the workshop is finished, if you can go over this in your own time, and you should be able to do it by your own, because Galaxy team puts a lot of effort to make these materials like very self-explanatory, so that you can reproduce, right? So there are links how to do that, like I just done it, right? Like I just gathered all of them into a single collection, and all these steps are mentioned over here. Like you select, then you from all selected, you just build the dataset list. So all the materials are here, so no need to worry. You can go about this or any other material from this site, which is like Training Galaxy, right? Okay, now our samples are ready in our workspace. Now the next thing is we want to do some quality check before proceeding with the analysis. And this is important again to get to know about our data, right? So, and this can be done using a tool called Nanoplot, right? So let's go and find that from our tool sheet. Okay, so this one, okay, I forgot to introduce to the whole platform. So this is basically called tool shield. Here you can find all different kind of tools. At this point, to best of my knowledge, there are about 5,000 tools already available in major servers, major UseGalaxy servers, right? And yeah, so this is the central panel where you can like edit your tool inputs and you can visualize the stuff, right? And this is the traditional history box, so where you can see all your history, what you have done, or like what kind of data you have stored, right? So let's search for nanoplot over here. So out of all those list of tools, I select nanoplot. So here, even to just to know like what a tool does, you no need to go to, like, most of the time you no need to go to the tool's individual help page, right? Because most of the time here, otherwise in the down section you can find that description. Even if you want to go to the help section, then you can find the links over here, or if you want to cite them, the citations available to read for the publication, right? Okay. Now, now as we combine them in a batch mode, all the samples, so we're gonna select this option called batch. But if you have like different samples, you can say combine, and you can select multiple samples, right? Mhm. Sure, sure, I can do that. Is it better now? Okay, thank you. Yeah. Okay. So now we are in the batch mode. And out of here, so for our files now, they are like FASTA files, right? They are not FASTQ files. So here from drop down menu, you can select whatever kind of file formats you have. For now, we have FASTA. And then here, what are the file modes? Like if you want to choose a single data set, like which is one FASTA file, or like multiple FASTA file, like you can select using your control plus click or something. Otherwise, now that we have this combined list, we can select this collection data mode, right? And here, this is the collection one. And simply for this tutorial purpose, we are not going with any advanced options. We're just selecting the by default options which provided by the tool, right? But if at all you want to see what are the other parameters being used, you can see it from this list, or if you want to provide, you can modify accordingly, right? Also, if it's a long running job, so you can just if you want to receive email notification after the job is being completed, you can provide that as well. So what you're gonna do once your job is finished, is gonna just trigger a email, which is very handy sometimes for a long running jobs, right? So now the last step is, after all the things or all the parameters are set, you just need to do execute. So it's gonna trigger another job over here. So yeah, so why this lot of outputs? So this nanoplot basically produce a lot of different things, and like HTML report, nanostat, and all these different files, right? And it need to do for how many samples? Like 6 samples, right? That's why you can see all these outputs basically, right? So yeah, so this is gonna take some time. Yeah, now from queue to now they are being executed. Yeah. Meanwhile, let's go to our material space and see what else we have. Yeah, if you see here, so this is the hand section parties, and here you can see all the steps we just did. Okay. Okay. Meanwhile, those are being generated. Let me take you through the what are the reports we're gonna see. So you're gonna basically see a HTML report with all the plots and all the statistics basically about the reads, whatever like FASTA files you have, right? So here you can see in one side read length and one side your number of reads, right? If you see, like most of our reads like coming in the range of like around, this is around 10,000 5,000 or something, right? So this is how can you determine like what's the like how your majority of the genes fall under which kind of length, right? This kind of small from things you can detect from this diagrams, right? And many more information to be sure. Let me show. Okay, these are done. These are very quick. So let me go to one of the HTML reports. I can show you. Okay, so yeah, this is one of the HTML reports. So how did I went? Like from history, you just need to find the proper start, like whichever start you want to check, and just go to them, go inside that, and you can see for all the 6 samples, you have the results, right? You just need to do view data. So this is a typical nano plot report. So here you can see summary statistics like mean of the length, like which is see even from this diagram you can see, right, the mean of the length is around like 5,000, which you just saw, and median read length and number of reads. So in these samples particularly we have 15,000 reads, right? And the read n50 value, which you can easily compare for your assembly. Then how many total number of bases are there in these samples, right? So yeah, so these are the all the essentials one should look for while doing quality control of the reads, right? So for scope of this workshop, like we are not going through details. You can of course read in the Nanoplot website about all these plots. So yeah, so these are the kind of reports you get for all your sequencing data, right? Okay, so the next step, like we saw, like our reads looks good and everything. Then the next step is we're gonna do the assembly. And before doing assembly, need to do pairwise alignment for them. So again, if you're coming from biology background, so you must be knowing so what pairwise alignment is basically. So it's just gonna, if you have two sequences, it's gonna match the each base pair, and it's gonna align on them, and it's gonna assign some kind of score based on your alignment gaps or different methods, right? So maybe you can read more about Pare's alignment. So this we're gonna do using a tool called Minimap 2, right? Which is also available from the tool sheet. And we're gonna choose Minimap 2. So here this comes option for Minimap, map with Minimap. Okay. So here as well, you have a lot of options. So what we're gonna do on the first step before the assembly, we're gonna do the pairs alignment with the same rates which we already have, right? So that means the same reads gonna map with the same reads, and it's gonna form a alignment file, right? So for that, we don't need an external index because we're doing pairs alignment with the same sequences. So you're going to get it from the history itself. History, that means which sequences we already have, right? Which is our like plasmid collection list. So again, from this thing, we're selecting plasmids, and these are single end reads, and against what we want to map, right? So here I'm going first because like I know these options, right? For you, you can of course you can read about them here, or otherwise you can go to the minimap site as well, right? So here I'm selecting plasmid, and now I'm selecting the second set of genes which we're gonna map against, and here those gonna be the same things, right? So we're doing pairwise alignment, plasmid against plasmid. That means all sequences against all sequences, right? And even here we are not selecting any more options. Let's see if we really need to do that. Okay, yeah. Here one option we need to select, like from which is like Oxford Nanopore all versus all overlap mapping, right? So for easy interpretation, like if you're doing it in from command line, you need to give a lot of options, which I can show you just in a bit. Okay, select this. Yeah. So here you can select a profile. So if you're doing it from command line, you can give this different parameters basically. But for here, like they are combined into single profiles, and you can provide that as well. from here, right? So we're gonna select Oxford Nanopore all versus all. Now, so what kind of alignment file we need for this purpose? Because the next tool we're going to use called Miniasm, and it expects us to have a file in a particular format, which is called your PAF file format, right? And we're gonna see what this file format looks like, right? So let's execute, then I can show you what this file looks like. Okay, now again, it's gonna take some time. Then before that, let's go and see what this path format is, right? And I hope you guys are doing it side by side. And if you have any questions, maybe you can ask. Uh, Ras, can you check if we have any questions for now? Like if they're struggling with any of the doings? Okay. Uh, if no questions for now, then I would like to introduce you to this map file. How does it look? So it's gonna have a sample name basically, and these are coming from your reads. Sorry, these are okay. Cool. We can go about that.
Yes, sir. We have first of all shared you the chat link. If you can, you can also visit there.
Okay.
I can read you out some questions if you like.
Let me see the question from chat section. I guess they are like blended with all the charts. So
Yes, sir. So I can reach it out for you if you want.
sure.
So, first question is from Rubaid Ahmed. He
Mm-hmm.
wants to know that to sequence via PCR, you need different primers. How many
Yeah.
primers would we use to get convincing result to detect pathogen via sequencing?
Okay, so about this sequencing like protocol, I'm not sure. Maybe it can get from mini website, or if Ansu know, maybe she can share. So about I'm really not sure about the wet lab like design of protocol of this one.
Yeah, so basically the point is that in most cases, and when you do a metagenomic kind of experiment, is when you would need to I do the pathogen identification, because if you are doing a isolation of a specific bacteria from the sample, you probably would already know what it is because you're using a specific media to grow it, right? In case of metagenomic samples, in most cases, what is being referred is like a 16S RNA is being sequenced, and then there are reference databases at NCBI or Codex, which is then referred for identification or basically taxonomic binning of these sequences. More recently, people realize that that may not be the best way. So there is something that you now try and do, which is called as an average nucleotide identity (ANI), which is a measure of nucleotide level genomic similarity between coding regions of the two genome to identify if they are similar or if what you have identified is a completely new, different taxonomy. So in terms of how many primers, so in most cases it is the sequencing of the 16S RNA. And if it is not 16S RNA, then what is preferred is a whole genome sequence. And in that context, what you calculate is an ANI to assign to basically identify what pathogen it is.
Okay. Thank you, ma'am, for the answer. So we have next question from Richa Shukla, and I guess that is again for you, ma'am. The question is, how could we use a human microbiome project for bioinformatics studies, say sequence alignment, as the submissions include 16S RNA sequences?
Yeah, I can see that is asked by Richa. So Richa,
Yes,
I'm not really clear on what you are asking. So you can use the Human Microbiome Project for several things. So if I can get more clarity on what exactly would you like to use it for? that will depend on the basically the approach will depend on the question that you are asking. So if you're listening to me, can you just rephrase the question, and I can see if I can answer it, because right now I'm not sure what you are wanting me to answer, because you can use the same project and the same data to answer several things. So I will wait for you to revert and tell me what exactly the question is.
Okay, I'm sure, ma'am. We'll wait for the question. Thank you, ma'am, for the answer. Sir, so we have one question from Ajay Sharma. He
Mhm.
wanted to know that can we do RNA-seq data analysis in a bed file format, or it is necessary to have data in FASTA file?
Okay, so generally, like when you sequence your this RNA sequencing, you get your results if you're doing it from Illumina or something. Most of the time you get in FASTQ format, right? And it depends upon your experiment, like how you want to design your experiment. Like you can do it in like pairwise sequencing, or otherwise you can do it in single end sequencing, right? So it totally depends from experiment to experiment, like how you want to design it. So I hope that answers the questions.
Uh,
If I just add to what Sangram said, so in case of RNSec, most people would now like to use a differential expression, right? You want to know how different the two comparison experiments are. So they use DESEC2. And in most cases, you can use BED or FASTA. There is no constraint because the file format conversions are available. And in fact, there is an entire tutorial on using DESEC2 for analysis of RNA-seq data within Galaxy. So that's there. Though it starts with the fast QC, and it actually starts with the way you are saying that do you need faster sequences? Yes, it does that, but there is no constraint like that.
okay,
Yeah.
okay. So thank you, ma'am. Thank you, sir. So our next question is, can Galaxy be used for other organisms like parasites to check for resistance?
Of course, yeah. Like it's matter of like having the files in place, right? Like, and most of the time, even for if you want to align to some kind of parasites, there are like inbuilt genomes are already available to align against them and do further analysis based on that. So yeah, this is just a platform to give you uh, give you the tools and the files you needed, right? And then the possibilities are limitless. You can do most of the stuff here. Yeah.
Yeah, so thank you, sir. So we have next question from Saroj. Like
Mhm.
for the specific disease, can we get the read count sequence data from Galaxy?
Yeah, for read count data, exactly. So if you have your FASTQ files, like then if you can process them in a way, like you have different protocols, right? If you go in a traditional way, you have Toxito pipeline and all of them. Then if you go through them, in the end, you get the this tabular format uh, read count data, right? Like you can go through Tuxedo pipeline, then you can go to feature count and all these tools or uh, ST6. So different tools are there which can quantify your reads basically, and then you get your read count data. Yeah.
Okay, okay. Thank you, sir.
Okay.
Ma'am, okay, so Richa Shukla just posted a question. I guess she deleted it again. So she's rephrasing the question. We'll wait for her. So, ma'am, sir, we have next question from Shani Malik for networking like Cytoscape. Which format can be used in Galaxy?
Uh, okay. For that one, like I haven't explored that part. So maybe I can come back to this question later. So, but what I know, there are tools which can explore network-based files, right? So,
Oh,
yeah.
okay. So,
Yeah. Yeah, so basically to answer this question, Galaxy actually works with Cytoscape, and in most cases they would recommend a JSON file as an input file format.
Yeah.
okay.
Yeah.
Okay, thank you, ma'am. Thank you, sir. So, sir, one of the question is like, if we are using our own data, how do we
Mhm.
select the proper settings?
Okay. That's a good question. So proper settings, you mean like I am assuming the proper parameters, right? For parameters for a particular tool you want to use, right? So for this, like it mostly comes from like reading the tool's description, and even going from like if someone already does the experiment using the same tool and what kind of parameters they use. At least for to when I select for particular parameter. Otherwise, it's most of the time like trial and error. Like let's say we're going for the next step right here for doing assembly, and over there are a lot of different parameters which depends to get a proper assembly, right? And that can be tried again and again to go to a proper optimized value of a particular parameter. So,
Okay.
Anshu, do you have something to add?
No, I think it's all right. I was just looking at the other questions. This one is okay because this. So I was looking at the other question in terms of Richa again asked. So I think her problem, really, what she was asking is because the sequences are sometimes very huge. How do you perform alignments? So yeah, so Richa, one thing is there are several ways of handling metagenomic sequences. There is one way where you first do taxonomic binning. The other way is you do taxonomic binning later on. First, and you do annotations first. So it depends on what your strategy is. But in most cases, what you are getting from a metagenomic sample, at least with the other NGS platforms, are short reads. So those short reads are either assembled in a way for you to give a contiguous stretch, and those contiguous stretch can be either annotated first and then put in taxonomic binning, and then you generate the whole organism, or it can be vice versa. You know, it really depends on what approach you are using. But what you need to remember is that every alignment that you are planning to do, you need to make sure which alignment tool you are using. Like, do you want to do an end-to-end alignment? Like you are actually using a global alignment or a local alignment? Are you looking at motifs? Are you wanting to annotate a specific gene? Or are you looking for homology? So again, depending on why are you doing the alignment will decide your experimental approach, right? So if you can write a mail to me on what you are trying to do, I can probably help you out with that. And to very quickly answer Firoz's question, how do we take care of the human genomic DNA sequences when you do a sequencing from blood? It's a very valid question, Firoz. 99% of the data that you get from a blood sample will be human and not a pathogen, right? And it's an open question still because we really don't know how to very human sample. So there are issues of still using metagenomics in clinic. In most cases, it's like you basically isolate the organism first and then do sequencing, which is not what we want. We want something that you can sequence from the whole sample and then go ahead and do annotations. So I would you just take a look at the current state of the art. It's still not a resolved issue when handling metagenomics because of the simple issues that the human genome itself, the human proteome or the pan proteome, as we say, is yet being identified. So the pan genome is yet being identified. So you really don't know what is that you need to eliminate. All right. So yeah, it's an open question in the field, Firoz, and if you can write an algorithm to really eliminate all the human samples data from these metagenomic things, it will be a new method. There is absolutely no doubt.
Okay. Thank you very much, ma'am, for the answers, and sir as well. Thank you very much. So I guess right now we are done with the questions, and so we can go back to the presentation. Okay.
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 Thank you.
Thank
Yeah.
you. Thank you, ma'am.
Yeah. So let's resume with the question. Sorry, let's resume with the analysis. Like if you have any more questions, maybe we can answer towards the end, right? So yeah, so where exactly were we? So we mapped all our reads against the same reads using minmap2, right? And it gave us a certain files per sample, right? And we wanted to output these files in a path format, right? And these are nothing but like if you want to explore this file format, there are certain columns, right? If you see, these are same file which we got even in our analysis, right? These are the same files. So if you see here, they have different columns, right? And if you can see, these are your like query sequences. Like different query sequences given a name, and those are mapped against the parallelement happen against another target sequence. So this is your target sequence, right? And then what is their length, or like, yeah, what is the sequence length, what is the start position, what's the end position, all of this information mentioned, and the relevant string and all these informations. So this is a particular file format which is can be taken by the next tool, which is Miniasm, right? And what it does like ultra fast de novo assembly, right? So of course, like with this kind of long reads, the error rate is high, but at least it able to give you a standard like assembly, right? And which can be optimized going further with other tools, right? So let's do that. So now we have something like this kind of alignment files, right? Now we have sequences and their alignments. Now we just need to build our assembly line, right? So let's find about minimap miniasm tool. Okay, so here it's asking about our sequence reads. So the sequence reads are the plasmid sequences which we already collected in a single group. And now it's asking about our pap samples, and which is like basically generated in the last step, right? So if you see here, there are like this is the rate, like these are the samples which are mapped against using MiniAptor, right? So you can directly select it over here. So now, do we need to select any advanced options? No, at this point we don't. So now let's simply execute that. So now this gonna provide us with a assembly file, or like what generally called assembly graph. So now this assembly graph, you can visualize this assembly graph. There are tools called bandage are available to do such kind of stuff, right? And your assembly file just looks something like this. You have a particular scaffold or config, then you just have sequences against them, right? And this can be actually mapped using bandage. So this is a tool called bandage. It have the desktop tool, but again you can do the exact same thing in a Galaxy itself, right? Let the files being generated, then I can show you how we can achieve this thing, right? So what is this assembly like? What's the benefit of visualizing this assembly, right? So here you can clearly say like how your assembly is being formed, or if they are very much segmented, or they are very much intact. All this information you can say simply by looking at the assembly from a visual point of view, right? And yeah, so once we get our assembly file, we can do that. Even this bandage tool can handle several outputs from several other tools. For now we are doing with Miniasm, which produce this graph file. But also, like if you heard about the sorted sequences, at some point you must have heard about this velvet tool, which helps you do de novo assembly of short reads. And spades, trinity, then mega hit, all these alignment tools can assembly tools can give you similar kind of files, right? And you can visualize this using bandage. And most of these tools, like I guess all of them already available in Galaxy, right? So yeah, you can just try them out. Okay, now these jobs are also being running. Okay, so now let's see what gonna be our next step basically after this. So at this point, we are very far fetched from, not very far from, like at least we have several more steps to go before we find the our antimicrobial resistance genes, right? So that's our goal, right? But so now what we have our assembly files. So now we have assembled our plasmid, basically you can say plasmid DNA, uh, from the sequences which we have done using this nanopore sequencing. Now, once we have that assembly, basically we can detect those genes from that assembly, right? So yeah. So the next step gonna be like yeah. So we just need to increase the accuracy of this assembly, right? Then there's another step called like remapping with MiniMap2. So whatever assembly we got, we just remap against this assembly we already got, so that So this is a repetitive process. So our assembly gets more accurate with these steps, right? So let's see. Okay, let it run for a few more minutes, I guess. Okay. Then once we have this, then we're just gonna do the conscious moduling using Recon, right? So here, what's gonna happen? So we have the our um, we have our assembly, right? Then we're gonna just refine it a bit more using Recon, right? And so for now, you are seeing this several step, right? So first we're done using mini-F2, we do it pairwise alignment, then we're doing miniism, then we're again constructing the assembly, then again we're remapping them. So there is an alternative tool to all of this as well, right? So there's a tool called unicycler. So I guess at this some point it is mentioned over here. Okay, I'm gonna show you the bandage like assembly visualizations as well. Okay. Okay, so here's a tool called Unicycler. So what it does, this Unicycler, inside them, it use several aligners and it optimize your assembly in a single go, right? So instead of doing this all these steps again and again, so you can simply give your sequences at a time, and this gonna do all these steps by itself based on a few parameters, right? So yeah, we can try that out parallelly so that it can at least go in a queue. So let's search for the tool. Yeah, create assembly using unicycler, right? So now here we can choose like the same files which we got initially. Uh, so these are the initial files, right? Which even we done the payload alignment. So here we can directly give those instead of any kind of alignment files, right? We can directly give our FASTA files, and then we just need to choose a few parameters. Let's see if we have any parameters to choose. No, right? Okay. So for now we are going with the default parameters basically, and we'll start execution. So yeah, so if you come with the description. Okay, let's go about bit about its description. So like I told, under each tool, basically you get most of the descriptions in Galaxy itself, right? So unicycler is a hybrid assembler pipeline for basically for doing bacterial genomes, right? Like here we're dealing with bacterial genomes, right? And it can take both long reads, short reads, like which are Illumina reads, and even long reads. So which you're currently doing for Nanopore and like some other stuff. So internally, it basically uses this Racon, which we are doing manually, step by step. But Unicycler already using these things, Minimap and Racon inside it, right? Let's see how far we with this thing. Sometime it's gonna be slow, otherwise it completely depends upon the your size of input file, even and the availability of your resources, right? But most of the time they are quite fast. Okay, so if you see for almost 5 of the files, the assembly is done, one is going on. Let's see how the assembly file looks. Yeah, so this is your assembly file. So they are sequenced with each single context, and some context might be very long, which sometimes is a good config, right? So now let's see how this assembly looks basically, right? Let's go with bandage tool. Okay, so this bandage tool usually takes this like assembly graph files, which are these files which you just generated. Let's give, yeah. So okay, no, we're not gonna choose the final assembly. we're gonna choose this assembly graphs which generated using minism, right? Even here we're going with default parameters, but of course you can choose the parameters depending upon your need, right? Okay, then this is gonna take some time because before this get executed, this things need to be finished because they are happening in a queue basically, these jobs. Okay, so meanwhile I can show you like what's the next step. So now we had the reads, we just done the assembly. Now the last step gonna be finding your this plasmids and even your antimicrobial resistance genes, which is the aim of the experiment, using few more tools, right? So towards the end we have two more tools like which are, yeah. So one is called, one is called Plasflow, which gonna help you detect your plasmids basically from this bacterial sequencing. Okay, and this uses like neural network to basically detect the plasmid sequences, right? And the next thing we're gonna detect is like your finally the antibiotic resistance genes, right? And this is gonna use several databases like your race finder, then plasmid finder, and strammer. Okay, so strammer is a tool. So strammer is a tool which gonna help us do the scanning of this antimicrobial resistance genes, right? Using these databases basically. Okay, so let us see, once our assembly is ready, we're gonna apply uh, these tools to our assemblies. Uh, do we have any burning questions which we can take? Meanwhile, these files are being processed.
Yes, sir, we can check for some questions. So, sir, we have a question from
Uh-huh.
Chitabjita. Sorry. How can we decide from the assembly visualization whether it is good or bad?
Yeah, sure. So let this tool get generated. I can directly show you like which can be considered as a good assembly. So mostly it should not be very fragmented, right? That's you don't want your assembly to be very fragmented. It should have a single assembly, and even based on different statistics, like you can see your like N50 value and different statistical point of values to determine if your assembly is good or not, right? So for validating this assembly, even there are other tools also available. um, to be precise, I don't remember the tool name right now, but I can suggest later on, like if they can. Yeah.
Yes, sir.
Yeah, just to add to what Sangram said, see, an important part here is we are trying to assemble plasmids here, right? And if you look at the visualization in Galaxy as in the workflow system, the unicycler or other things are actually giving you, like if you see here, they are trying to, or Maniasm, they are trying to give you a shape of what they are seeing. So the whole point is a plasmid needs to be circular. So till the time you get an assembly which looks like a circular assembly, you visually know that probably the arc on the top that you see here is not a good assembly because it's not looking like a plasmid so far, which means the assembly is incomplete. There can be unresolved sequences, when then that is why you cannot actually close the loop. So a good assembly is something that if you try align sequences using the overlap, the hanging overheads to a, on both sides of the sequences, and it gives you a complete circular shape. In this case, you would know that the assembly is very good because you can actually manage to generate the whole plasmid. And likewise, in genome assemblies also, one way of using visualization is to see how much of your reference genome is getting assembled. Not the ideal approach, because we now know after the the observation of the pan genome that not all strains will have the same genetic component. So there can be different strains within the same species that can have additional gene components. And so doing a reference assembly might sometimes will have unmapped reads that you just ignore, but they are part of the sequence as such. So the point is how big a contiguous stretch one can make after an assembly is over or indicates the quality of the assembly, and it is good if you're, as Sangram was saying, NGA50, NG50, so Quast is a tool, QUAST.
Exactly.
If you give your sequences to Quest, it will actually pre-compute all these values for you. It will show you based on a reference. So it does both. It does reference-based assembly. It does de novo assembly. So based on what you are doing, it will tell you how many times in your sequence there is a misassembled region, and whether the base call was of good quality or it is not believable. So you actually get a lot of these things, and I would recommend you some of the new people who are doing genome sequencing to play with MAUVE. MAUVE. MAUVE is again something that even Richa can use for doing alignments, which are like more whole genome based, end to end, and it really gives you a very neat idea of synteny, how the sequences are moving across two genomes. So it actually allows you to understand big chunks of genomes being, how they are aligning across two different genomes. Because in case of at least bacterial sequences, there is a very strong structure to function correlation. So where does a gene occur in the genome has to do with the function of that particular gene. So if the position of that particular gene or operon has changed, it also indicates acquiring a new function or modification of the same. So that's why MOV is something that you can use if you are not working with like 1,000 sequences. If you're working with 5 or 10, it's a good idea to do assembly or the contig ordering with MOV, and then use tools like Quast. The other tools one can use to look at the visualization of assembly in case of organisms which show very high genomic rearrangements is Siberia. So that is again you can use either command line or online. So these are the applications in which you can do an assessment at your end of if you visually see I can see that my sequence look good in terms of assembly, they are good, and all of these tools give you NGA50, NG50, and several other numerical parameters to judge the quality of the assembly, and not just based on visual inspection, but the actual statistical measure that you can apply on a larger data set that you cannot manually evaluate.
Thank you very much, ma'am, for the answer. Thank you, sir. The next question is, can we do virus genome assembly using unicycler?
Uh, so this is yeah, I hope yes. Like I haven't read much about this thing, but yeah, this is specially designed for like bacterial assemblies. So of course, like you can use for, but I guess there are other like accurate viral assembly assemblers are available.
Okay.
Yeah.
Thank you, sir.
Yeah, and just to add what Sangram said, see, viral genomes are very, very small. So if your nanopore starts to work correctly, you practically have just a single readout, and it's out there. And unicycler is, as Sangram is saying, it is primarily built with the objective of circularizing the plasmid data so that you know that it is complete. So it's a very specific case here that we are dealing with something that completes the cycle. But in case of viral genomes, in any case, they are very small. So I really don't see a very big issue in assembly when with this long rate platforms that are available now.
Yeah.
Yeah, thank you, ma'am. Thank you, sir. So my last question would be from Femi. He wanted to know that is there a place we can find tutorials on how some of the tools in Galaxy can be used?
Uh, yes. So there are like vastly online tutorials are available, but ideally like you can go to this site for most of the training materials basically. So it's just training.galaxy.org, and here you can find different range of topics, right? Let's say if you want to do based on sequence analysis. So yeah. So yeah, once you go here, like you can see like for mapping based things, then variant analysis. There are several tutorials like using prokaryote genome. So yeah, so this should be your site to visit to explore different tutorials.
Yeah, thank you very much, sir. I'm sure they would go to Galaxy training for any further, like anything they want to know on Galaxy. So we can continue with the presentation. So I guess we are done with the questions as of now. Yes, sir.
Sure, sure. Yeah.
Thank you.
So even yeah. So even our data are being processed. So that's a good point to start again. So yeah, so this is the visualization, right? Like what you're just saying. Even if this is uncomfortable to see here, you can also download. Okay, so I haven't told about other great features which you have here in Galaxy, right? And this is a good time to say about those. So if you want to just download them, you can simply download them over here, right? If it's a PNG file or it's a tabular file, like last I shown you, even if it's a FASTA file, you can download them to your local system as well, right? Also, Galaxy provides you with a lot of different options. Like say for now, all our files are being named with based on our fast file, right? The first file, which is like the faster file. All the files are being named like that. If you want to rename it for your convenience purpose, then you can do that using this edit attribute, right? Even there are some other details available. Let's say this is a image file, but once I go to a tabular file, I can tell you like how easy it is to filter a large tabular file, right? So yeah, so these are the small options, but again, this makes your life very easy, right? Even now, let's come to the this documenting part, right? Like so, you now you're executing all these tools using just click and click thing, right? But what if someone like some of your colleagues want to do them using command line tool, right? Then in that case, like all this metadata what are being executed in background are being saved. So you just need to click on this detail button, right? So if you click on detail, yeah. So here, each step by step, everything is being documented. Let's say like what is your version of the bandage tool, and when they run, like how the standard output looks. So I guess okay, this is the image file, so we don't have any standard output. But even if you're getting some errors, you can capture the error as well, uh, even a lot of different information. So for now, we are doing it point and click. But I guess if some of you have attended previous sessions of this, then you must know like someone told about this. It can be accessed. The Galaxy platform can be accessed using APIs as well, right? So yeah, you can programmatically do all these point and click things from programmatically as well using Galaxy's BioBlend APIs, right? Even so for now we are using default parameters, right? And each of the parameters are being noted down. So how you generally do when you do command line, you just note it down, right? Like yeah, so here this much parameter options I have chosen, but you no need to do that here, because here each and every parameters are being documented, which is a really great thing. And finally, this is the command line which is being executed. So at certain point, if you're being comfortable with the command line and you want to switch to command line, then what is next step? You just need to copy paste these commands, right? Like with supposingly you have this file in your locally, right? And these are the parameters which you're just seeing over here. So it's that easy, and each and every step is being documented over here, right? So yeah, that's the good thing about this Galaxy platform, and it really follows the principles of like fair principles, right? Like the fairability, documentability, like the shareability of platform. Yeah, about shareability, I'm gonna come towards the end. How you can basically share your whole analysis to any of your teammates or any of your collaborators. Even you can provide in a publication as well, right? And that really legitimates your analysis, right? So let's go with the remaining steps. So now you got to know, right? We can basically see the visualize the assembly basically. And as Anshu said, you generally expect like plasmid assembly to be like very much circular and not this kind of fragments, right? The different fragments. So this is out of six samples. This is one assembly, but yeah, there are others as well. Once you go through them, you can visualize them individually. So even our final assembly also done using which we have done using unicycler basically, and this seems to be our final assembly basically. And we can do our post analysis on this, like basically calling out the plasmids and calling out the antiviral genes, right? And for that we need to go to the tools we're gonna use. Yeah. We're gonna use call something called Plasflow, right? So this is using neural network to detect the your plasmids in your assembly, right? So here we just need to provide the our input gonna be the sequence it's gonna be the final assembly which we just created using unicycler, right? And execute. Okay, now it's gonna generate like several files, right? Like all your chromosome details, all your plasmid details, so and some additional files, right? So even, yeah, that's a good thing. Even the intermediary files are being saved here. So at any point, if something is being failed, you can really check where exactly you got failed, right? Here we don't have any failed results, but I can show you once this is over, I can show you a failed result and how you can detect the what's went wrong, right? So now parallelly, while this is being executed, now let's run the antimicrobial antibiotic resistance genes as well. And for that, we're gonna use the tool called Starmer, right? So let's search for that. And again, this tool also gonna take our the final assembly, which created using unicycler. So this basically scans genome assemblies against these databases, right? Race finder, plasmid finder, and point finder databases for AMR based genes, right? So again, we no need to give individual files. Instead of that, we can give the our final set of files, which is the final assembly. So if you're just seeing this one collection, don't misunderstand with one single file. This having 6 files in them, right? So if I go 64, right? Yeah. So each time you create, they are uniquely assigned with a particular identifier. Like let's say this step is called 64. So you can actually go back and check, right? So yeah, so see, this is the 64th one, and it is a list of 6 files. So these are the individual files, right? And once we're running this timer, it's gonna run on all these 6 files while we're selecting this one step, right? And now just execute it. So yes, now Samner also gonna give us some results, right? And this gonna be the expected results, and this gonna take some time. So meanwhile, let's see like what other features we have, right, with Galaxy. So like I said, you can share the entire workflow of your system, right? So yeah, so if you're seeing here, you are using this 4% of the data. So I'm using about 10 GB of the data. So see, just with free quota, you can use up to, I guess, 500 GB of storage. So that's well enough for doing a standard analysis, right? But if you require more, like you can, of course, you can always install the local version of this in your institute or in your local system as well, and you can proceed with the analysis, right? Okay, now let's see. I just want to share this workflow with someone, or some of my colleagues, which they want to replicate it or just want to see the results, right? So what I can do, I can go to this options menu, and here there's an option called share or publish, right? So just go over here and make the all this history as a accessible via link. Like you can either you can invite individual, yeah, either you can invite individual users by using the email ID, otherwise you can make a public link, right? And now if I just share this one with someone, they should be able to see this kind of page where you can see some of the metadata and the results. Let's see. Okay, so this step also is done. Now let's see the results. So you shared this one with someone and they can simply click over individual results to see basically. Okay, this is Excel file, but yeah. So I guess this seems like a log file which is being generated from the Starmer tool. Let's go to a particular file. Let's say I want to go to race finder. Okay, race finder results, which is done. So yeah, this is our final file basically, which gives you like a lot of columns it have, but it ideally have these genes. These genes are present in our samples, which we sequenced like we came from the FASTA files, right? We assembled them, then we detected these things. So these are coming from those sequences basically, and ultimately those samples. And these are the genes which are like antimicrobial resistance, right? So now you have your genes of interest. Now it's upon you, like how you want to go and find more information about that. So with this tutorial, if you see, they have we have suggested, right, there is this database card, right, like comprehensive antimicrobial resistance antibiotic resistance database. So here you just need to search for a particular tool, like we have detected several genes, right? So let's say we want to go with this and search it over here and go for looking more information about that. So here you can go and see all the details about this particular gene, right? And the very comprehensive informations are mentioned about this particular gene of your interest, right? So this is how ultimately we have done. We found the antimicrobial genes from a few FASTA files, which is generated in a from nanopore sequencing, like which is like very fast and efficient, faster way and cheaper way basically. So yeah, that's the journey, finding antimicrobial chains using Galaxy. And alongside, you saw different of these advantages, which really helps you do reproducible analysis, right? And also, yeah, I forgot to mention one thing. Let's say you have done this analysis, right, and someone want to repeat this analysis, right? Then you can simply share this workflow. Like if you building a workflow, you can simply share. Otherwise, let's say I already have a workflow. So here is the option, right, to workflow. So I already have a workflow. Okay, so this workflows, if you see in tutorials, they have already provided some workflows. These are nothing but a text file, which is with GA format. And this can be downloaded once after you built your own workflow, right? See, these are the all the steps. If I can minimize, I can show you better. Yeah. See, we started with this thing, right? Like the plasmid IDs, like all the input samples basically. Then we done a QC using nanoplot. Then we done assembly using like miniasm basically, like after doing minimap. And then we also done, oops, maybe some, yeah, then we use the bandage tool to see the how the assembly looks and all these informations, right? So this is a really nice workflow which is shareable basically. So you can simply save it or download it and share it with your friend, right? And the next time you comes, like let's say for now you've done a test run with a few of the samples, and you want to do it with large number of samples, right? And that time you don't want to sit and do each steps manually by finding these tools. So once you have this workflow, it's very easy. You just need to run and need to provide your initial inputs. Like in our case, it is a list of FASTA files, right? So let me just create another history, give its name like from workflow, and here I just need to give particular input file, which is our plasmid list, right? Okay, so I just need to give this one input, and because I don't need to manually like change any of the parameters. But if you want, you can, of course, you can change the different parameters of them, right? Like here you can change individually all the parameters. But for now, let's run with with the default options. Then you just need to do simple run, and it gonna trigger all the steps for you from the starting to end, whatever you saw. Okay, whatever you're seeing over here in this galaxy, like after these things executed, you see a small dialog box over here. So these are the really exciting people from Galaxy, which are doing a lot of open source projects. And meanwhile, your analysis is being queued and jobs are being executed. You can do this lot of fun stuff, right? So here, this is a really nice project. They're trying to do a machine learning, and here they're asking your help to build the model basically. So identify this, which is like male and female. So yeah, these are the small interesting gems of about this galaxy. And as you can see, all the steps are being scheduled, like there are 64 jobs. So all the steps basically which we went through step by step just before this. And they're gonna triggered, okay, they are being triggered in the another history. Oh, I should have checked the history earlier. Okay, so if you can see here, we have done up to this, right? That we used a strammer to find out our genes. And after that, see all the steps again being triggered from the same workflow. And all of these steps gonna happen automatically. And if you provided a UML ID, once this is done, you should receive a, the workflow is finished. So yeah, that's with all advantages in doing analysis using Galaxy. So thank you from my side.
Thank you, sir.
So,
Thanks,
Neha.
Sangram.
Yeah.
Yeah, it's really nice. So Neha, what I would recommend is there are not many questions on the chat. So
Yeah.
is it okay with you? I'll finish my part and then go to Q&A.
Yes, ma'am. Sure, ma'am.
All right. So I think what Sangram has managed to convince us is that Galaxy is a pretty useful platform when it comes to identifying of AMR genes. But you might have also realized that how hard it is to understand antimicrobial resistance. Like some of you who have been hearing it for the first time, it's pretty difficult. And let me tell you one thing, without the engagement of everybody who is involved, like everybody who needs to take an antibiotic, we are not solving this problem of AMR by just doing research. There has to be other verticals that we need to initiate. So what we thought that it will be interesting and also useful if we can convince our next generation about the challenges of antimicrobial resistance. And in order to make them understand what it is, we have developed a video game. So you play the video game to understand how antibiotics work and how antimicrobial resistance develops. And it's a strategy-based game. So what we are doing is the game is ready for launch, and we are looking for beta testers. So I'm going to play a 1 minute video now that is going to introduce you to what the game is about. And then there will be links in that video for the beta testing, and I'll even in my slides afterwards. So before I go back to my presentation, I would like to show you. Let me know, Neha, can you see a blank screen?
Yes, ma'am.
All right, so I'm playing the video now. Let me know if there are any problems. Okay. And for all the viewers, so for viewing the video in a best possible way, kindly make your screen full screen and volume can be a little higher. So yeah, here we go. Yeah, so this is basically to introduce you to the fact that we are not just trying more scientific ways, but also trying society awareness ways on generating awareness on antimicrobial resistance. So can you see my slides now?
Yes, ma'am. We can see it.
All right, so just very quickly on the game. So the purpose of this game is to generate awareness among citizens of tomorrow. And all levels in the game, so far we have 8 levels, are based on sound scientific principles. So we are not boring the community of knowing what they are, but till the time they understand it in a strategic way while playing the game, you cannot go to the next level. So it's a strategy based design. We have 6 game levels in Skin World, 1 level for lung and COVID-19. The game is already endorsed by WHO India, and we are trying to see to how many people we can ensure that they play the game and learn, because learning happens when you do something over and over again. So we thought that if the game is hard enough that you need to play it several times, you will learn about the concepts of AMR, and the game is Armor, and it was recently covered by Times of India. So Armor is currently supported by the Jigyasa virtual platform, CSIR India, CRI France, and Sangram University of Paris, and WHO. So we have a very nice support for the game to develop it further. And the features and learning strategy, some of you who are gamers out there, we have followed the tower defense game genre to basically design Armor. It has 2 worlds, skin and lung infections, and we will be introducing more tissue type infections as we go forward. And the skin world has 6 levels. Each level has 1 learning message, which is integrated into the game mechanics. Overall, the message that you get after playing the game is that both overdose and underdose of an antibiotic will lead to drug-resistant bacteria, and even a mild wound that gets infected can prevent it. Point is, how do you gamify this whole concept? So we have done that. So the game is available for you to play. The interface of different levels look like this. So yeah, I'll not go into the details because I don't want to kill the fun of you finding how it works. And these are the URLs for you to register for the beta testing. We are looking for beta testers. I'll come to the game, the links one more time. We introduced the lung word and the COVID-19 level for several reasons, because the TB is, as you might have seen several places now, that it's a bigger monster than the ongoing COVID-19 pandemic. And in COVID-19 level, we are conveying the message that antibiotics don't act on viruses and only bacteria can be killed by them. So we are using the current pandemic also to make sure that people don't misuse antibiotics when not needed, and it's a very, very big issue, and we would really like to sensitize the wider community. So it's an invitation to the beta testers. For some of you who don't know what a beta tester are, so beta testers are real users, as in all of us. And in this case, we are targeting school-going kids, but not necessarily that. Everybody in the next generation who can test the game and share their feedback, identify bugs or issues, and make a recommendation. Say, for example, a beta tester can be somebody who tell us, I enjoyed playing the game, or I learned something. You really need not be technical, but we want our beta testers to be very, very responsive. So if you are interested in beta testing the game, we can send you the game link. What you need to do is to go to this URL and register. We are just asking for your very basic information so that we can send the game URL to you, and you become one of the first people to basically play the game and give your recommendations. So with that, I thought we will close the AMR component of the workshop. And in next 10 minutes, as I told you that we will be taking examples from Galaxy, I'll show you a few more examples of where we have been using Galaxy in my lab for a very long time for several different things. Since we took one example on antimicrobial resistance, I thought we'll take the next example from mitochondrial rare diseases. Just to give you in context that it is not just that for one specific activity you can use Galaxy, one can use it for several different purposes. So to introduce, I'm pretty sure all of you know what a mitochondria is. So in every cell, except a few cell types, and I'm pretty sure you know which cell types have mitochondria. And then these mitochondria, in every cell type, there are several mitochondria that are there. And in each mitochondrion, there can be several mitochondrial DNA. Now, the mitochondrial DNA itself encodes for 13 protein coding genes, 2 rRNA genes, and 22 tRNA genes. But the majority of mitochondrial proteins for its function are encoded by the nuclear DNA, which is almost 7,000 proteins. So you know that there are like 13 protein coding genes coded by mitochondria, but all the genes encoded in mitochondria are responsible for a very large number of diseases, and I'll show you quickly what it is. Initially, when we were sequencing mitochondrial DNA in early 2000, it was mostly for phylogenetic studies or for forensic applications, not so much for medical research. But of late, if you see that the green vertical is, as you go from 2003 to 2015, you see that the number of genome sequences that are available in public domain are growing more and more for medical research as well. The reason is that in 2016, the Mitochondria Medicine Society recommended next generation sequences as the first line of diagnosis of mitochondrial diseases. So it is no more an optional thing. One has to do this in order to do a proper diagnosis. And this is what I was telling about. It's simple. Mitochondrial DNA is a circular DNA. It's a small DNA of only 16 kb in size. But if you see the number of disease associated with each gene on mitochondria, you can see how many are those, and I'm not even listing all of them. So diseases in mitochondria are mutations in mitochondria lead to several mitochondrial diseases because mitochondria is core to several functions in the cell, like apoptosis, lipid, nucleotide, amino acid metabolism, the electron transport chain, which actually makes mitochondria the powerhouse of the cell because you generate ATP. So the point is that we were to understand which of these mutations are actually disease causing in mitochondria, and my lab has been developing several platforms in terms of prediction models, databases, and visualizations for many years. I will not be describing this to you. It is basically to convey to you that even from within the same lab, when we are focusing on the same problem and developing different resources, at some point we were stuck when we were to integrate it. Like say, for example, I'll show you this. MidSNP score was developed with a simple PHP based architecture. MitoLSDB was developed with LOBD platform, and MidBrowse was developed on a JBrowse platform. Now we were thinking that it's better to integrate all the three applications because one is a database, one is visualization, and the third one is a prediction method. And we were stuck because there was no way to put all of them together in a fashion that it can be useful it can be used by anyone. So imagine this was resources developed from within the same lab, and despite the fact that we followed standards, sometimes they just don't talk to each other. So that is when we actually landed onto Galaxy, and we call this platform as MitoLink. It has databases that are centrally shared and shared data libraries. It has all the tools like Query Builder. It has a large number of variation analysis tools, not just from our lab, but from other labs as well. It has a large number of converters and mappers. And as you can see, from data to analysis to visualization, we have managed to capture all because Galaxy allows us to generate APIs and to allow it integration of all these applications. More recently, we came up with the standard guidelines for mitochondrial DNA variant interpretation, which will also show up in MitoLink very, very soon. And this specification guideline is also approved by ClinGen. So this is a very important contribution that I work with a large number of researchers and clinicians from across the world to develop these guidelines for interpretation of mitochondrial DNA variant, and MitoLink will soon have this information. And this is to just show you that despite the fact we were developing many, many resources over years, it's only through Galaxy we managed to put all of that together as a single window interface to the community. Now, not just for NGS data analysis, Galaxy can also be used to understand or to do drug discovery. Now, this is a platform that we published in collaboration with several other labs called Molecular Property Diagnostic Suite. And again, no need to do coding or programming. A large number of early stage drug discovery applications and visualization are already available on this platform, and one can use them. So with that, I would like to basically conclude the part of giving examples of Galaxy. So we looked at examples of NGS, we looked at examples of AMR, mitochondrial diseases, and very, very quickly how in drug discovery also one can use Galaxy as a platform. So the platform is like an envelope, a framework. What you would like to put that as tools and workflows is completely decided by you, and it's the flexibility it offers makes generation of these workflows pretty flexible. This is basically to tell some of you may not know where Institute of Microbial Technology is. We are based out of Chandigarh geographically, but if you look at our contribution to the global databases and prediction methods, we rank among top 4 institutions in the world in our contributions to mathematics. So this is a site where you can go to see how many databases and how many countries they evaluated. So we made huge contribution and we would like to continue to do that. Personally, in my lab, I work with a very large number of undergrads, school kids, post-graduation, master students, a lot of them, and we publish together. The reason being very simple: when you work with a wider community, you are thrown open with several challenges and it gets you learning all the time. One of my favorite projects is this one because the first author of this paper, Pooja, was just finishing her 12th standard when she published this paper. She ultimately went on to become the Gates Cambridge Fellow and now she's doing her PhD in York University. So this is basically for all the students out there. You really don't need degrees to do science. What you need is passion and motivation, and I think that is very, very critical. So just follow your passion is the message. And this, as Yasha was telling you in the beginning, I work with a very large set of students because the data-driven projects, the sheer amount of data, you just don't need computational methods. You need enough human brain at work. And that is why you need so many people to put their brain in solving so many problems that we work with. And just to acknowledge what all I presented today is coming from my lab in India and in France. And I would really like to also thank my co-speaker today, Sangram, for agreeing to take you through Galaxy. It's really nice of him to agree to share with us his experience. And finally, thanks a lot. Thanks to the DTU team for having us here. And if there are students out there who are looking for PhD positions, I am looking for passionate PhD students in my lab right now. So if you are there and if you're willing to join the lab, I'll be very much happy to work with you. So with that, I would like to thank everyone for listening to us for so long and would be happy to take any questions that you might have.
Yes, ma'am, we have a few questions. So, ma'am, one is from Ajay Sharma. I think it's regarding Galaxy that what to do if we have a big experiment data. I think Galaxy offer 250 GB, and here I have approx 350 GB of RNA-seq. So, is there any medium to process this kind of data in Galaxy or any other platform?
See, what I would recommend is that have a local installation of Galaxy. So if the data size is huge, the recommendation would be to get the tools to the data rather than the other way around, because it's going to take a lot of time to get the data to the tools. So follow that in principle. Second is if you have RNA-seq data, and I'm not sure if you're comfortable working with the Tuxedo suite or the DE-Sec2. Most of these codes are available with R package. So you can actually try an alternative, and even within R you can use Galaxy. So Galaxy allows you to integrate your R programs in the workflow system. So recommendation would be to have a local Galaxy and try and do your analysis using those R packages, and then use the Galaxy interface to talk to the data in a GUI-based fashion.
Yeah, thank you, ma'am. So the next question is from Aparna. She wanted to know that can Galaxy workflow be constructed for every package in Bioconda or Bioconductor, or are there any exceptions?
I really cannot answer that, but because the reason is it's very difficult to say all. They do provide that, but Sangram, would you have anything to add here?
Yeah, sure. So, so most of the tools, like which you can see in Bioconda, they can be put it into a tool sheet. It just basically you need to do some configuration based on Galaxy's recommended way of configuration. Then yes, of course, you can pull them into your tool sheet of either local installation, or if you want to install them in a public server, then you can request the Galaxy team from respective site. So yeah, so to answer your question, all the tools you can find in Bioconda platform, you can put them into Galaxy.
So thank you very much, sir. for the answer. Thank you very much, ma'am, for everything, for the presentations and everything. So, ma'am, please, Yasha is there, and I guess you'd like to say something, ma'am, please.
So I would only like to thank Dr. Anshu and Mr. Sangram for their extensive hands-on on Galaxy. And I'm sure on one hand, while the participants would value collaborative research that Dr. Anshu has been doing for years now, they would also be able to acknowledge the power of Galaxy, and they would also be able to use it for a reproducible and transparent computational research. So thank you very much, Dr. Anshu and Mr. Sangram.
Thanks, Yasha, for having us here. And if I can request you, I can share the link for the game. And if you would be willing to share it with your participants, if somebody has not managed to follow the YouTube link, just for their benefit.
Sure.
Thank you,
Thank
Yasha.
you very
Thank you,
much,
Neha.
ma'am. Thank you, sir. Ma'am will share your link with the participants in the YouTube channel. And now, everyone, thank you, everyone. So, thank you, participants, also. It was a great experience. And now we would like to conclude our session. And just remember that we have our last session at 2 p.m. IST, and then we will have our valedictory session.